Started. And I'll get started by muting someone. Thank you all for joining today's Florida Coral Reef webinar. We have some excellent experts here with us today. Christy Kerrigan, Sarah Fangman, Joe Unsworth, Alex Newfield, and Penny Cutt, all of which will be introduced to you shortly. We will be on this webinar for approximately an hour, and don't worry, there will be plenty of time for questions. So just going over some quick meeting etiquette. Please keep yourself muted. Really, the only people who should be unmuted are the people who are presenting at the time. After a little while, we will be having questions, but please put your questions in the chat. The webinar will be recorded, and the recording of the transcript will be available later. I just understand that Commissioner Beamfur has arrived, so before I move forward, I will actually let him say a few words because he's been such an excellent champion of the reef. So Beamfur, I'm going to, oh, you're unmuted. You have the floor. Thank you, Commissioner Fur.
Hi, how you doing?
Excellent.
You got a great turnout here today. That's wonderful.
Yes.
Yeah. Just a real quick note, just to welcome everybody, and just to let everybody know there's some actually good things going on that we are presenting to the legislature this next session. 7 counties have gotten together and are working on having the EPA and the Florida Wildlife Commission work on the box that was kind of put around the ocean reefs as a conservation area. We're also going to rename it. We've asked for a rename in honor of Kristen Jacobs. So we're seeing some good work there between those 2 agencies to develop a management plan that's critical. And if we can get that going, we'll have made some major inroads. So I'm glad you all are here. There's a lot of good information coming up, and thanks for the invite.
Thank you for being here.
Yeah.
And I forgot to mention, you're wondering who's probably speaking here. I'm Alyssa Jones-Wood. I am the Green Initiatives Coordinator for the City of Hollandale Beach. I forgot to introduce myself. So back to the meeting etiquette, just please keep yourself muted. Only presenters can share their screen, so don't even try. Ask questions in the chat box, and we will answer the questions at the end. And again, this webinar is being recorded, and the recording and the transcript will be available afterwards. Wanted to make a quick mention. Thank you to our sponsors. This webinar is part of a series of webinars that was actually meant to be an in-person event in March of last year before COVID-19 hit. So we're trying to make the best of it in a virtual situation. And Hazen was a large sponsor, Pelican Harbor Seabird Station, Pinnacle Ecological, Nova Southeastern University, Helmholtz College of Natural Sciences and Oceanography, and our National Parks Conservation Association all sponsored the Ocean Day, and thus this as well. And then thank you to our organizers. This event and all the next ones coming would not have been possible without the major organizers of the City of Hollandale Beach, the City of Hollywood, Broward County, Florida Department of Environmental Protection, Friends of Our Florida Reefs, and Sierra Club of Broward. So thank you. I hear someone is unmuted. If you could please mute yourself. We have other upcoming Ocean Day webinars, one every month until about March. So the next one is January 12th. That'll be focusing on marine wildlife. And in February, a low nutrient landscaping and gardening webinar. So you can get more information on that at floridareef.org/oceanday.ht.html. And last bit, just want to introduce our moderator for today, Penny Cutt. She's the director of Friends of Our Florida Reefs. She's been in South Florida since she was 6 months old. By day, she is a director of Cummins Cedarburg, a local coastal engineering firm, and in her free time, she's an avid boater, diver, and recently started a Coral Reef aquarium. Her master's work was at Nova Southeastern in marine biology and coastal zone management, and she's a former U.S. Army Corps of Engineers, South Florida Water Management District, and Miami-Dade County Environmental Regulator. Penny is also appointed member of the Broward County Marine Advisory Committee, Pompano Marine Advisory Committee, and Pompano Beach Zoning Board of Appeals. So thank you, Penny, for joining us and for moderating today's discussion. So without further ado, I'll hand it over to Penny and start Christy's ability to share her screen. Thank you.
Thank you, Alyssa. Definitely excited to be here and to see such a huge turnout. Thanks everyone for spending your lunch learning about our Florida coral reefs. Appreciate that introduction. but the stars of our shows are actually our presenters, and we're going to start out with Christy Kerrigan, who's the Assistant Manager of and Reef Resilience Coordinator for the DEP Coral Program. Christy, we're excited to hear about the first time you went snorkeling and diving on Florida's coral reef system. So why don't you enlighten us to your first experiences here with our reefs?
Yes, thank you. Can you guys hear me?
Yes, we can hear you.
All right, thank you. Yeah, so my first time diving was actually the first time I got my job here with the Florida Department of Environmental Protection, which was about 5 years ago. So I was diving with the incredible coworker and staff along my side. So it was pretty incredible. And it was, I believe, in Miami-Dade County. So. Right, shall I kick it off, Penny? Can you guys see my screen?
I can see your screen, but I think it might be a second monitor.
Okay, yep, all right, let's see. How about
There
now?
we go.
I'm going to set a quick timer to try to allow time for everybody. So I'm just going to set a timer for 10 minutes, give you a heads up when we're getting close. You want a 1 minute or 2 minute warning, Christy?
Sure, 1 minute sounds good.
Okay.
All right, thank you. So I guess good afternoon. As Penny introduced, my name is Christy Kerrigan. I am the Assistant Manager of the Coral Reef Conservation Program within the Florida Department of Environmental Protection. So I'm going to kick off this panel with a high-level overview of Florida's coral reef, where it's located even. I'm going to highlight our disease outbreak and some of the response efforts, and then sort of touch on what the state is getting into in our restoration world and restoration efforts. So I'm going to definitely try and stay within the 10 minutes. Oh, here we go. I'm trying to click. Okay. So giving you a map of Florida's coral reef. So it does extend down from the dry Tortugas all the way up to Martin County, and it's about 380 miles of reef. It's adjacent to one of the most densely populated and highly urbanized coastlines in the world, and more than a third of Florida's population does live along our coast. But as you do move from south to north along the reef, management of the reef does vary from federal to state agencies. But I did just want to hone in our region, which is the northern third of the reef. And
Did others lose Christy's audio as well?
yes.
Yeah, I think everyone did.
Looks like her screen is catching up. Christy, we lost your audio.
It's important to point out that these reefs are just hugely important. It provides a home to thousands of organisms, including corals, gorgonians, sponges, fishes, and other invertebrates. And not only that, but economically, the reef provides 71,000 jobs and generates over $6.3 billion in sales and income. In terms of coastal resilience, the reef is our first line of defense against extreme weather, providing shoreline erosion and coastal flooding. So in a recent study that was done by USGS, they reported that just 1 meter loss of reef would result in over $356 million in damage to infrastructure along our coastline. And so with the 6 million residents and 38 million visitors a year, you could say that these reefs are hugely important. Unfortunately, though, we are moving into a time where we're starting to see more and more decline, not just here in Florida, but around the world. We're starting to see these shifts in what normal even is anymore as these systems continue to change and degrade. I'm sure many of you guys have heard about the mass bleaching events in the Great Barrier Reef a few years ago. Well, these same things are happening here in Florida. But not only that, here we do have such a dense population, which does create a lot of human pressure. And so with that, water quality is a huge stressor. We have fishing pressures, damages from anchors and vessels, marine debris, and these large coastal construction projects. But not only that, Florida has been experiencing this ongoing coral disease event since 2014. And we've been seeing these major declines of many of our primary reef building corals. These are the corals that are primarily responsible for driving Florida's economy and providing that first line of defense in terms of coastal protection and providing a habitat for fisheries, et cetera. So researchers are still working to identify the pathogen or pathogens causing this disease, but we've been collectively calling it stony coral tissue loss disease, or SCTLD. But here in the map, you'll see this outbreak started again in 2014, right in Miami-Dade County, and it started spreading north and south. So by the end of 2015, you'll see that it's made its way through Broward County and down and through Biscayne National Park. Jumping ahead 2 years later, so by 2017, you'll see that it's made its way all the way to the northernmost extent of the reef tract and down into the Middle Keys. And so where we are today, here we go. Where we are today, you can see the disease is now essentially through the Marquesas. And the only place where this disease has not hit is the dry tortugas. Hope to keep it that way. So as you can see, this disease is spreading really fast, but fortunately we do have a lot of incredible people and partners helping to respond to this effort. And I just wanted to throw this slide up with this logo party here. These are all of the really great partners that we have from agencies, universities, NGOs that are all working collaboratively in this collective response effort. And so I want to thank all of them because it wouldn't be possible without everyone. And I'm going to show this slide, but not spend a whole lot of time. You can see that this is sort of our disease response structure, and we broke our response into different response teams as sort of these different elements coordinating. And I just want to, for today, highlight the work of our coral rescue team, propagation, and the restoration trials team. But we do have a lot of other folks working on different elements like data management, communications, intervention, et cetera. So the coral rescue team, so their main mission, and they started a few years ago, was to go out and quote unquote rescue the corals in advance of the disease outbreak. So basically protecting them before they become infected. And so we're bringing these, removing them from the system, putting them into land-based facilities with thanks to a great partnership with the Association of Zoos and Aquariums. So these corals are now living in 5 facilities here in Florida, but then also 10 facilities in other states throughout the country. So that's really exciting, and we have a lot of partners with that. And so after, of course, these coral collections are completed, and so we start looking at land-based and in-water propagation of these colonies. And not just of the rescued corals, but also corals from what we call the endemic region, which is part of the reef that has been exposed to the disease outbreak. And so we could call these corals survivors, and we can also maybe safely assume that they have some kind of resilient characteristics that are important. And so we also need to look at bringing these corals into these land-based facilities. But to do that, we do want to make sure that we start building up the infrastructure so we can be ready to start scaling up restoration in a meaningful way. And then that brings me to the restoration trials team. And before we are able to really scale up outplanting, we need to first understand whether the corals that do get outplanted become infected with the disease. And so the restoration trials team designed a pilot project to understand just that. So partners up and down Florida's coral reef will be outplanting corals in this replicated fashion to see if they survive and to see if there are any impacts to the surrounding reef. And so this experimental study will be kicking off next summer, and we're really excited to learn a lot of great things from it. But that effort is really just focusing on restoration in the face of disease. But one thing that we can say for sure is that Florida has been a leader in restoration and outplanting for over 15 years. And so we have groups like Moat Marine Lab, Florida Aquarium, Coral Restoration Foundation, University of Miami, you're going to hear from those two in particular today, have all been conducting restoration and have been really successful with it here in Florida. And since the disease outbreak, a lot of these groups are switching their focus from the acropora corals, which are not susceptible to stony coral tissue loss disease, to growing more of those mound and boulder coral species that we've lost so much of and are critical for providing those ecosystem services. And taking it a step further, these groups are starting to look at restoration of the entire ecosystem, not just corals. So grazers like our diadema or sponges, etc. And that's a really important element too in looking at resiliency of the ecosystem.
Just a heads up, we've got less than a minute left.
Okay, thanks. So this is just a map showing locations and regions of the different partners up and down Florida's Coral Reef for all these different and sort of their region of focus. So we've got a lot of folks out there working on this, and it's really important. But in terms of planning, as Coral Reef managers, we do want to make sure that we're doing this work in a smart and strategic way. And so really to start scaling up these activities, it's important that we have a framework that lays out the site-specific goals and objectives. This work has actually already been done in the Florida Keys National Marine Sanctuary. I know you're going to hear from one of our other panelists about Mission Iconic Reefs. But for us here in the Coral ECA, we're a little behind the curve, but we're hoping to kick off a process soon. And we hope to model off the incredible work that has been done in the Keys. But then in thinking about Florida's Coral Reef holistically, it's important that we have a strategy that unifies both of these plans with high-level goals and objectives for the whole system. And that's another process that we hope to be kicking off soon. But ultimately, I just want to point out, what we want to do is restore the system's natural resiliency. And so protection and restoration is going to be our top mission over the coming decades. So I'm out of time. I know that was very quick and very high level, but I'll be happy to answer any questions after our panelists and presenters. So thank you very much.
Thank you, Christy. And we do have some questions, but I think we're going to save them to the end. We've got them coming in through the chat feature here. And in the meantime, let's introduce Joe Unsworth, who's a research associate at Rasmus. And we want to hear about Joe's first time diving on Florida's coral reefs.
Ability, I remember, was probably about 30 centimeters. You could maybe see to the end of your arm, and that was about it. But in spite of some of those kind of like initial hardships, I do still find myself doing science underwater occasionally. And that's today what I'm excited to show you a little bit of the work we've been doing here at the University of Miami. Hopefully over the next couple of slides, kind of the ideal you'll get is how coral reef restoration can be kind of leveraged as a tool to conduct research and outreach. And so our lab is called the Benthic Ecology and Coral Restoration Lab. We're based at University of Miami's Marine and Atmospheric Science campus right here next to Biscayne Bay. And we've been conducting coral reef research and restoration in the Miami area for over a decade now. We started back in 2009. We have 2 offshore coral tree nurseries. Tree here is just referring to the structure that the corals hang off of and grow subsequently very fast. We also have a state-of-the-art indoor facility that's dedicated to growing coral on land. And we're actually in the process of building out a second outdoor facility for the same purpose. Both these facilities use filtered seawater that is pumped in directly from Biscayne Bay, giving the corals the ideal kind of growing conditions that they need while we're still constantly able to monitor them and take care of them. And so this map shows all of the sites in Miami that we've conducted our restoration work. we kind of stand at the intersection of being not only practitioners of reef restoration, but also students of it. So our organization has always prioritized research over doing pure coral restoration. So rather than simply aiming to outplant as many coral fragments as possible, we also want to gain a better kind of understanding of coral restoration and how corals interact with their environment. And what this allows us to do is explore new ways to kind of improve the coral restoration process, basically looking for ways to make it more efficient and maximizing the impact that our efforts have. And one of the projects that I'm kind of excited to tell you about today is called 100 Yards of Hope. This project started last year, actually, as a partnership between our lab and Force Blue. And Force Blue is a nonprofit that we've worked with before. They retool ex-military combat divers for the purposes of doing marine conservation work. Their recruits are extremely mission-oriented people. So being able to give them a set mission to be a part of is not only therapeutic for them, but it's also very productive for partners like us, because we get all of the help that they give us. And the original idea of the project was to restore a football field sized area of reef, hence the name 100 Yards of Hope. And this was amplified in addition with the Super Bowl being held in Miami last year and in Tampa this year, or next year, I guess. So this year, thanks to funding from Florida's Department of Environmental Protection, the project was actually expanded to include a whole suite of partners who are all kind of tackling different aspects of coral reef conservation. This project really allows us to do some pretty interesting disease work, whether it's still kind of unclear now that the disease front has passed through the Miami area, whether it's okay to go outplanting corals that may still be susceptible to the disease. So obviously we don't want to be outplanting corals that will just quickly succumb and die. So we're taking kind of the cautious approach in undertaking this project. So we've already surveyed the surrounding area for signs of the stony coral tissue loss disease. These surveys have confirmed no positive signs of the disease. We didn't see any signs of disease in the area. So the next step will be to outplant 750 coral fragments of susceptible species into the area. These fragments are corals that were dislodged during storm events, ship groundings, or were removed during construction projects. So we call these commonly corals of opportunity. They're corals that would otherwise die. So we're basically giving them a second life. And these are also what we would call non-naive corals. They're from the zone where the disease has already kind of swept through. And these are kind of the survivor colonies. So in theory, they were already exposed to this disease, and either they weren't affected in the first place, or they were affected and they were able to survive and recover. So after the corals are outplanted, we'll continue to monitor the plots for signs of disease. And if we observe any signs of what could potentially be stony coral tissue loss disease, our partners from Nova Southeastern University, Brian Walker's lab up there, they're going to kind of intervene and prevent it from spreading. And a lot of these different interventions include this antibiotic paste, different types of epoxy that kind of create a fire band around the disease margin to prevent it from getting to the rest of the colony. And these are kind of disease intervention techniques that we're also strongly utilizing in another one of our projects. This is probably one of our most ambitious projects that our lab has undertaken. It's the Southeast Florida Coral Reef Restoration Hub. And this is a project that's funded by the National Fish and Wildlife Foundation, or NIFWF. It includes performing interventions on over 1,000 of these colonies that are showing signs of active disease in both the Broward County and Miami-Dade County areas. We also plan to use fragments that have been grown in our nurseries to reskin colonies that have already succumbed to the disease. So you plant these nursery-grown fragments on these old dead colonies, and theoretically they fuse together and are able to skin over that dead skeleton to give the coral new life. This project also includes a whole number of other non-disease related things, including introducing these algae-eating sea urchins, the Diadema antillarum urchins that Christy mentioned earlier, introducing them to our restoration plots. They eat algae, which competes with coral. So theoretically, adding sea urchins to our restoration plots will make the coral have a much easier time growing and becoming huge, which is the idea. We're also going to be simulating how a restored coral reef reduces wave energy using the University of Miami's Sustained Wave Tank facility. This is a multi-million-dollar facility that the University of Miami has that's capable of simulating up to a Category 5 hurricane just 100 feet from where I'm sitting right now. And we're also testing out what's called managed relocation of corals, so moving them between different habitats and temperature regimes to see if this can help them survive future stress. So as you can see, all of these projects that we're undertaking take kind of a holistic approach to coral reef research. We're approaching the different issues that reefs are facing from a very wide variety, a lot of different angles to find out what works and why. And that's kind of reflected in the number of partners that we're kind of involved with. So each of these partners plays a different role in growing coral recruits to disease intervention, all of these different things. One thing that I haven't quite mentioned yet is kind of the critical role that people actually play in this coral conservation equation. And that's why we have Rescue Reef. So Rescue Reef is our lab's public-facing citizen science organization. What is citizen science? Basically, citizen science is just involving members of the public, citizens, in scientific work. So in addition to giving presentations and tabling events, we seek to actively involve members of the public, such as yourselves, in our research. So Rescue Reef holds expeditions where everyday citizens join us for a day on the water. So after a brief training in the morning, everybody piles onto the boat, and we head to our nursery where the volunteers help with maintenance, cleaning the trees that I mentioned before, our other nursery structures. And they also help us to collect the coral fragments that we then head to the nearby reef. The divers outplant the corals. They're able to use the same methods that we use in our lab's restoration work. We are purely there for supervisory reasons, just to make sure that they're doing the right thing. We demonstrate, and then they basically take the reins. And so since 2015, when the program was started, we've had more than 700 citizen scientists join us in outplanting over 5,000 corals. And we've actually compared corals that are outplanted by our lab's team members that we do in our typical field days, comparing them with those that are outplanted by citizen scientists. And what we found is that there is no difference in survivorship between them, suggesting that citizen scientists are effective at conducting coral restoration work. So basically, the citizen scientists who had only a couple hours training and a little bit of hands-on training as well, they did just as well as seasoned restoration practitioners who have been doing this for 10 plus years. After and before the expeditions, we also conduct surveys to kind of assess people's knowledge, attitudes, and perceptions of coral reefs. And what we see is that people take part in our expeditions. They not only leave being more knowledgeable about coral conservation, as they should, because we've been talking to them about it all day. They also end up caring more about coral reefs. So they're more likely to kind of advocate for environmental goals purely because of their experience with us. And that's really why we believe that citizen science is so important. Having these members of the public be kind of coral champions when all we can do is do the science and say that coral reefs are important. Having members of the public such as yourselves going out and doing that is even more critical, especially now as we're kind of facing coral reef decline here in Florida and around the world. That's pretty much all I have. I hope this has been a really informative look at what our lab is all about and the kind of work that we do. utilizing coral restoration as a tool for studying the environment as well as educating the public. If you're interested in learning more about the things I've presented on today, you can follow us at Restoration Hub and at Rescue Reef on all the social media platforms. You can also email me. My email is down in the bottom left-hand side of that slide. Thank you all for taking the time to tune in, and I'll hand the mic back to Penny. Thanks.
Thank you so much for that presentation. We appreciate your efforts and all that you've done to help others, help you, help the reefs. With that, I think we're going to go back to Joe Unsworth. And he's research associate at, actually, no, that was Joe. I'm sorry. Wait.
Alex.
Joe.
I can go again. It's fine.
Sorry, Alex. Alex, are you there?
No worries. Yep, I'm here.
Okay, and we can hear you.
Excellent. We're off to a good start.
Yeah, this jumping around's got us a little bit confused, and nobody wants to hear me today. Let's hear you talk about your fondest memory diving on the reefs, and we're excited to hear your presentation. And thank you to everyone for your continued patience.
Yeah, thank you very much, Penny. So my first experience actually diving on Florida's Coral Reef was back when I was in high school. I was fortunate enough to take a trip down here, and as part of that trip, my classmates and I took a snorkel boat from John Pennekamp Coral Reef State Park, and we got to go and snorkel on one of the reef sites down here. And I just remember pretty vividly this massive Goliath grouper that we found kind of tucked under one of the big coral bommies on the site we were at. And of course, I have no idea what site it was, because I didn't really envision myself at the time coming back, 10 years later to work in the Florida Keys. But now I like to think that maybe I've sort of swam by that specific spot once or twice since I've been back here. So that's kind of neat to think about. I'm going to leave my camera off in an attempt to preserve internet bandwidth here, but if you want to see my face, there it is on the title slide. Again, my name is Alex Neufeld. I'm the Special Projects Coordinator for the Coral Restoration Foundation, and you can see my email listed there as well if you'd like to get in touch with me after this with any questions you might have. So I'll just go ahead and jump right into it. I'm going to be talking about my organization today, Coral Restoration Foundation. We are a Florida Keys-based nonprofit organization that is dedicated to actively restoring the coral reefs in both Florida and around the world through some of our knowledge sharing. And our core mission is founded on 3 pillars. The first one is restoration, so doing the actual work of putting corals back into the ocean, back onto the reef sites where they used to live in abundance. The second pillar is education, so teaching people about what we're doing and ways that they can make an impact for our coral reefs in their daily lives. And then the third and final one is science. So while we are not strictly speaking an academic organization in the same way that the University of Miami is, for example, we do try and incorporate science and the latest in coral restoration and coral conservation in everything that we do. And we also hope that some of the things we've found in the field will help further the coral restoration work that's being done elsewhere. So how do we do this? Well, Joe actually touched on a couple of the strategies that are very common throughout coral restoration and conservation, but I'll dig into them a little bit more here. CRF's primary focus is with growing and then outplanting the 2 species of Acropora corals that we have in the Caribbean. And those are the Staghorn coral that you see on the top there, and the Elkhorn coral, which you see in the middle. And then we also have, in the last couple of years, worked on building up stock and outplanting techniques for the boulder corals. There are 2 species that we work with, and one of them is shown in the bottom image. So the 2 species of Acropora are actually listed as critically endangered on the IUCN Red List, which means they're just 1 step away from extinct in the wild. And estimates sort of vary, but we have lost somewhere between 95 and 98% of these corals in Florida in just the last few decades. So it's really critical that we work to restore the populations and sort of mitigate the ongoing decline that we're seeing. Now, CRF works with a total of 11 species in our nurseries. The other 7 that I haven't mentioned are at this point sort of just mitigation species, but we are looking to sort of bank them and then develop techniques for growing them more rapidly and then outplanting them eventually in the next few years or so. So I mentioned that we grow our corals in offshore nurseries. We have 7 of them, and they utilize the coral tree that CRF developed and pioneered about 15 years ago now. And together, these coral tree nurseries can grow somewhere between 45,000 and 50,000 coral fragments every single year. And what you see in this picture are those coral trees, and each tree consists of a unique genetic strain, or what we call a genotype. And these genotypes, some of them no longer exist in the wild because they're donor colonies that we originally collected from some 10 or 15 years ago have gone extinct in the wild. And so our nurseries represent the last remaining sort of instance of the genetic strain and those specific genetics that are really critical to restoring a population to its former glory. So in that same vein, we have designed our coral tree nurseries to harbor specific areas that we call genetic banks. And these banks serve to preserve the genetic diversity of the different species we work with. So while in some parts of our nursery, we may be, producing large numbers of corals and then outplanting them, the banks are sort of a safety deposit box for this genetic diversity. So we'll keep the corals there, we'll monitor them and maintain them, keep them free from disease or algae competition, but by and large, we want to leave them be so that we always have the largest swath of genetic diversity possible. And this is really important because, as we've seen, not just from marine conservation, but terrestrial conservation and restoration as well, genetic diversity is one of the key components in determining whether a conservation or restoration effort is ultimately successful. So to that end, we work with over 300 unique genotypes or genetic strains across those 11 species of coral. Now, in terms of the restoration side of things, CRF has historically outplanted on 30+ reef sites throughout the Florida Keys, all the way from North Key Largo down to Key West. But since 2019, we've really started to focus our efforts on 10 to 12 of these sites. And you can see in the upper left corner there, the 10 reef sites that we outplanted on in 2019. So in the nursery, once the corals have grown to what we call a reef-ready size, which for the staghorn coral is, somewhere between the size of your hand or both of your hands put together, maybe a cantaloupe-sized coral, and for the elkhorn, it's maybe the palm of your hand. Once they're that reef-ready size, we'll group them by genotype, place them into crates and bins that are full of water, and then transport them by boat directly to the restoration site. And once we get to the restoration site, it's up to our team of divers to use hammers and a 2-part marine epoxy to attach the coral to the reef. The hammer serves to just clear away any sand, algae, potential, nuisance species competition for these corals, and get down to that hard white limestone rock, the hard reef rock, which is what the epoxy will actually attach to. And after about 45 minutes, that epoxy hardens from a putty to a cement, and then the coral is firmly affixed to the reef, and it can start to grow down over the epoxy and eventually cement itself. So the clusters that we outplant in are placed pretty close together so that after all of the restoration is done, what you get is this blanketing effect across the reef. And if you can see in the bottom picture there, one of our dive boats, a whole team of divers working in a very concentrated area so that eventually as these corals grow and mature, they will become the dominant life form on the bottom, which is what we want to see in a restored coral reef. And we know that it works because, like I said, we use science to inform the work that we're doing. And so we're constantly out here monitoring these corals after they've been outplanted. And what our data show is that CRF's outplants have an average survivorship of about 77% across all sites and species. So 1 year on the reef, 3 out of the 4 corals we outplant are still there. And a C or C+ average may not sound like a lot, but when you factor in CRF outplanted about 35,000 corals last year alone. That's a significant amount of coral that's there on the reef that wasn't before. So we feel really, really good about this. What this image shows actually is a photo mosaic, which is one of the ways that we're able to track such large-scale restoration efforts through time. Photo mosaic is just a collage, essentially, of individual smaller photos that stitch together to create a very high resolution image that we can then zoom in, like you see here with these pullouts, and monitor our corals digitally on the computer instead of having to spend time in the water with things like rulers and paper and pencil. which works for a few thousand coral colonies, but once you scale up, it really becomes time prohibitive. Additionally, a 2016 study published by NOAA, and specifically Dr. Margaret Miller, revealed that in areas where CRF restoration was taking place, staghorn coral populations actually increased. And in areas where restoration was not taking place, the populations decreased. That doesn't sound like a super meaningful conclusion. It's sort of a, well, duh. But it's really important because it shows that we are making a difference. And if we were to leave these areas completely alone, not only would there not be coral there, but the corals, wouldn't be able to restore themselves. It essentially shows that we're at the point now where active restoration is required to not just increase populations, but just to simply sustain them. So it was very validating for us to see that our work was making a meaningful difference. These are a couple of healthy staghorn clusters that you see. They're about a year old on the reef, and each one of the colonies you see in these white circles was originally about 10 smaller fragments that grew up and fused together into one big colony. And you can see as you look towards the background of this image, more and more clusters kind of dotting the reefscape. And that's this large scale blanketing effect that we're trying to get with our restoration sites. The last thing that we see that really gives us hope is that our outplanted corals actually spawn. So corals reproduce sexually about once every year, and they release their gametes into the water column, and the gametes fuse and create new embryos that can then settle out and form new colonies on the seafloor. And spawning indicates that the corals we've outplanted are actually really healthy, because if they weren't healthy or they were too stressed, they wouldn't invest that energy in developing the gametes and releasing them in synchronization. And so what CRF has done in the last decade or so is returned over 140,000 corals to the wild. And you can see specifically these two images that are really, to my eye, very hopeful. A section of Carey's Fort Reef where we outplanted elkhorn corals, and the growth spurt that they showed between about 9 months post outplanting and 3 or 4 years post outplanting really shows that this kind of restoration is possible. The corals can grow and they can thrive on the reef still. We just need to put them out there, stabilize the population decline, and then sort of wait for them to take off again. So this bar graph just kind of shows CRF's progress through time with the different species of coral that we work with. And this is number of corals outplanted each year since 2009. And you'll notice a couple of things here. First, that the Orbicella, the boulder coral outplants, have really ramped up in the last few years, and we're looking to continue that moving forward. And you'll also notice that in 2017, we took a little hit because of Hurricane Irma, pretty much wiped out the back quarter of that year for us, but we responded with a big surge in 2018. And then obviously 2020, the COVID-19 pandemic has resulted in a slight decrease from last year in terms of overall outplants, but similarly, we're hoping to bounce back even stronger in 2021. So what that looks like then, just to wrap up, we're going to be working on incorporating a few new propagation techniques in our in situ nurseries. So that's new ways to grow some of those newer species really quickly so that they can be ready for restoration in the coming years. And we're also going to be focused on some experimental work, both in our nurseries and with upscaling our outplanting methods. So coming up with new ways to get more coral out onto the reef even faster than we're already doing it. And what this means is that we're hoping to restore some 40,000 different coral colonies across those 4 species and across 9 restoration sites. And those 9 restoration sites have been very specifically chosen, and they will include all 7 of the iconic reefs that we're going to hear a little bit about in just a minute. So to conclude then, I'd just like to, again, thank you all for bearing with us today and for joining us on this. If you have any questions, again, my contact information is there. You can also head over to our website, which is www.coralrestoration.org. We have, in a normal year, tons of opportunities for volunteers to work with us, both in person and occasionally from afar. We also run dive programs, which are similar to the University of Miami's Rescue a Reef program, in that we can actually take certified divers out with us to work in the nurseries and work on the reefs. And of course, there are always other opportunities to get involved, make changes in your daily life to help the corals. And all that information, again, is on our website, coralrestoration.org. So thank you very much for tuning in, and I look forward to answering some of your questions.
Thank you, Alex. I appreciate your patience and your presentation, and hope that some of our audience does take you up on your offer to volunteer with the organization. With that, we're going to move on to Sarah Fangman, who's our superintendent of the Florida Keys National Marine Sanctuary. And you remember the first time, I think this is you snorkeling in the Keys. So we welcome your presentation, Sarah. Thank you for bearing with us through this.
Certainly. Just want to make sure you can hear me.
Yes, we can hear you.
Yeah, we can hear you.
Fantastic. And then second check is that you can see my slides, because I'm not seeing what you're seeing. I'm just seeing Penny's smiling face.
You're seeing your slide. If you go to the top center where it says view everyone, you should be able to adjust that.
Okay. But you're seeing my slides.
We are.
Okay, great. Well, it doesn't matter. As long as you're seeing it, I can keep going. So yes, this is me kind of a long time ago, experiencing a coral reef for the very first time. I was not in Florida, but in the Caymans, and I will never ever forget what it was like to see a coral reef for the first time. I was a girl from Minnesota, so that was very unfamiliar to me. And people who know me know that for me, this was not only important because it set me on a course for my current career, but it also drives what I do every day. This experience that you're seeing me have right there as about 11-year-old girl. I remember that magic. And I know that girls and boys who are strapping on snorkels at this young age today are seeing a very, very different reef system than I did when I was their age. And so it drives me every single day to work to try to help these reefs recover and restore, so that when those kids are my age, instead of telling the story which I tell, which is I saw those declines that Christy mentioned at the very beginning of this this presentation, that kids these days are going to tell the opposite story. When they're my age, they're talking about how these reefs have recovered. And the previous presenters, and they're talking about restoration, I hope indicates that it is possible. So I know we're short on time, so I'm going to try to move through these pretty quickly and talk about what we're doing in response to those declines. And it has been noted, we've been seeing declines on our reefs and other reefs for decades. And I just, again, want to confirm that the slides are advancing. Penny, can you nod if they are?
Yeah, absolutely.
Okay, great, thank you. All right, so as we've talked about, stony coral tissue loss has added significantly to the declines we're seeing, but thankfully we have one tool in our toolbox to address that, and that is restoration. And as has been already talked about, this is something that has been pioneered here in Florida, and we are so very lucky that that is true. So the work that you heard about has been really impressive, inspiring, and I would even argue heroic. Unfortunately, however, to date, we're not keeping up with the declines. And not only that, in some cases, even if we were to wait for Mother Nature to, if we could eliminate all the stresses on our reefs, in some cases, the species are so far apart, the individuals that are left, they couldn't successfully reproduce. So even if we could eliminate all the challenges in our reefs, in some cases, these corals can't recover without active restoration. And so we realize that we've got to do something really bold to take it to the next level, to take the restoration work that we have done and others have done to date and really make it scale up to the point where it is ecologically significant. Now, some of this audience may be saying, well, wait a second, how can we be doing restoration when we haven't eliminated water quality problems, when we still are going to have hurricanes, when we've still got coral disease? Well, the answer is, number one, not every species that's out there is susceptible to the coral disease that's going on right now. And so we can work with the resilient species as well as the resilient individuals, because there are individuals of the species that have been affected by this disease that are still surviving and thriving, and we need to propagate more of those. And so it was this idea that we've got to do something, we've got to do it big, that launched this idea just over a year ago. December 9th of last year, we launched this effort that we called Mission Iconic Reefs, which was an attempt to address declines at 7 specific sites. And here they are in the Florida Keys. Some of them you already saw on Alex's slides. These are the 7 sites that we have identified because we think these are places where restoration has a really good chance of success. And so what we're doing, and I'm going to talk just really briefly because of time, what we're doing that's a little bit different from previous restoration efforts. Again, we're standing on the shoulders of those efforts, but we're kind of trying to stretch it and take it a little bit further. So what you're looking at here is a map of one of the 7 iconic reef sites, Sombrero Reef, and the different colors represent different reef habitats within the the reef itself. So not all areas of a reef are the same. And as a result, when you think about restoration, you would think about different corals that belong on those different areas. And so what you're seeing in this slide is sort of a sketch of a reef and the different habitat zones within a single reef. And you can see that we propose to do outplanting of different species in different percentages based on what would naturally occur there. And so the elkhorn and the staghorn corals that Alex and others have been talking about, those are the ones we use a lot. But you can see there are additional species that we are proposing to put into the mix here. Not only that, we are proposing not just to outplant more corals, we certainly need to do that, not just more species, but we're also proposing, you'll see in the middle there, to supplement with grazers. And it was mentioned earlier, and I apologize, I couldn't hear all of the previous panelists because I was struggling with my own connection issues. But I know it was noted that there are grazers that have a really important role to play. So the Mission Iconic Reefs is not just about outplanting more corals in greater numbers, but also supplementing that with grazers to help those corals have a better chance of succeeding. So right now, if you see in gray, when we launched this, the sites that we've selected have about 2% coral cover. And you can see, you look all the way across through time, our goal is to recover about 25% coral cover at these sites. Now, that's not going to happen overnight. It's taken a long time for our reefs to decline, but we are striving to get there through this ambitious project. This slide itself could be a whole hour talk. It's really exciting, though. I just want to mention that right now, not only do we have ambitious goals in mind, but there is tremendous advancement happening in the science and practice of coral reef restoration. We've got new species being outplanted. We've got new techniques that will help us do this faster. As has been noted earlier, some of the outplanted corals are starting to spawn, which is exactly what we want them to do. So there's tremendous amount of hope happening in the realm of restoration. And so this Mission Iconic Reefs effort is not just to sort of create these pristine reefs where we've done recovery and sort of set them aside. We want to get people involved so that people can come and see them, and so that they can witness what is possible and help support it, but also get involved. Earlier in this panel, I saw a chat asking about, well, how can people get involved? Well, we're absolutely developing programs so that divers can come and help us with this. So stay tuned for more on that. We have a lot, as you can see from the statistics, a lot of ambitious plans. And I'm really pleased to say that a good amount of funding has been already raised in just a year. And that the number you see there, $9.5 million, is not a complete. It's the estimate of those funds that I'm aware of. But because this project is being done with a lot of different partners, they're receiving funds and seeking funds that I probably don't even know about. And speaking of partners, we don't have quite the logo party that Christy showed for the disease, but this is some of the key partners that got this Mission Iconic Reef started. And we continue to build partners for this, but a lot of folks are behind this. And I think that's part of what I'm really hopeful about this mission, is that we have, all these organizations have been working in this realm historically, but now we're really doing it together. And so that I believe as a result, we're going to be even more effective. So our hope is that, like I said at the beginning of this, young people who are starting to explore and experience reefs, when they are my age, they will be able to speak to the dramatic recovery that they have been able to witness. And so that is our goal, to have these reef sites become, once again, iconic, healthy, thriving ecosystems. So thank you very much for your interest. Sorry I went through that really fast, but I was trying to be sensitive to time, and my email address is on the slide. Website has more information about the mission, and thank you so much for the opportunity to quickly tell you about this project.
Thank you, Sarah. We really appreciate your time and everybody's time as we go over a little bit here with the issues and glitches we've had on our webinar. We're hoping that moving forward, our future webinars that we've got planned for January 12th and February 1st won't have these glitches. Not quite sure what happened there. We do want to thank Hallandale Beach for making this happen. The city of Hallandale Beach has sponsored Oceans Day for us, and as this event couldn't happen in person, we're grateful that they were able to make this work for today, for this webinar and this format, and all of our presenters for being so adaptable. Let's see. We do have questions coming in from the audience, obviously. Christy, are you still with us?
Yep, I'm here.
Okay. Can you talk a little bit about some of the groups, the appreciation and awareness and fishing, diving, and other uses, and what some of the groups with DEP are trying to do to help? We've talked a lot about corals, but we're getting some questions in through the chat on people wanting to know what else is being done to help the reefs from other perspectives other than disease and restoration.
Yeah, sure. So Penny mentioned these focus areas that we call them, and we have 5 main focus areas that our program really hones in on. And so we have the awareness and appreciation focus area. And so that's like our education and outreach. Michelle Graltes is actually here on this webinar today. She is our awareness and appreciation coordinator, and she runs a lot of our education programs. We work with a lot of the elementary, middle, and high schools in developing lesson plans for them. This is part of the Teacher Trunk program. We have in our fishing, diving, and other uses focus area. And so that is really targeting our fishermen, divers, our commercial, you know, all those folks. We have a couple of different projects that are ongoing specific in that realm. Nothing so much that engages the public, but then our reef injury prevention and response program, we have a coordinator who helps coordinate responses to anchor damage. So it is under the Coral Reef Protection Act. It makes it illegal to anchor on corals, and so we help to track different incidences, go to those sites, and monitor and assess the damage that has been done on those reefs, and then reach out to those folks and sort of work with them to remedy the situation in different ways. And so our other focus area is Reef Resilience, and sort of under the Reef Resilience umbrella, we do have a couple of citizen science programs that are a great way for folks who are listening in to get involved. We have the Cfam program, it's the Southeast Florida Action Network, which is a citizen science reporting and response program that is really designed to improve the protection of our reefs offshore. So it basically is a mechanism for folks to report different marine incidents, so marine debris, if you saw coral bleaching, coral disease, vessel groundings or anchor damage, invasive species, etc. You name it. If you feel like there's some issue happening on the reef, please report it, and we sort of work with the appropriate agency or organization to respond in different ways. And that is a really easy website. It's cfam.net. You can go there to get more information. If you wanted to take it a step further, you can participate in the Bleach Watch program, which is really focuses on coral bleaching and coral disease in particular. And you go through this 2-hour training class. And that website is cfam.net/bleachwatch. But those are really great programs for folks to get involved. And yeah, I was rambled on a while there, but I'll pause.
Well, I'm really glad you mentioned the Bleach Watch training, because we did get several questions about how other people and divers can get involved with what's going on and help. And that Bleach Watch training is an ideal opportunity. And when we can, we do, in addition to the classroom training on Bleach Watch, we also take the divers out on dive boats and do field calibration to ensure that what you learned in the class you can apply to the field. And then we encourage you every time you're diving to record your observations and upload them to the Cfam website so that we can collect that citizen provided diver. It's very similar to, a lot of people may be familiar with the Reef program, where citizens collect fish information and identify fish on the reef. There's also an app for exotic invasive species where lionfish and other invasive species can be reported. So we do appreciate the citizen divers that are helping contribute to all of these opportunities, as well as the opportunities that our speakers presented on how you can help with some of the outplanting of the corals that are being propagated by some of these groups. What else do we have? We have anybody that'd like to ask a question directly? Unmute yourself and ask if everyone doesn't go at once.
Can I ask that we touch on, there were a lot of questions about beach renourishment and the impact of this imported sand and renourishment projects on the reef. Can someone speak to that, please?
Sure, I'll take that one because I don't know who else would want to. When we nourish our beaches offshore, we either utilize nearshore sand that's found between our reefs or upland sources of sand. And in Dade and Broward counties, as many of you know, we're out of sand that meets the state sand specifications. It's either too close to the reefs because we have to have suitable buffers to ensure that we don't end up with sedimentation or turbidity affecting our reefs when we dredge it, or the sediment characteristics are not appropriate. They've got too much fine material or too much rock material mixed in with that sand, so it's not beach compatible. And so more and more we're using sand from sand mines, generally located in Central Florida, which are historical shorelines of Florida. So when Florida was a stubbier state, a stubbier peninsula, those were historical shorelines of Florida. And if you dig down, you can get to that beach sand, and that sand is actually ideal because it's already been sifted and sorted and stratified by grain size by the wind and waves when those shorelines were created. And so a lot of that material is being trucked down to our shorelines here. And I know there's a lot of people that are looking to find other ways to get sand on the beach, but the reality is we've got our offshore sand, we've got our inland sand, but unless we're going to allow our barrier islands to migrate naturally by removing the buildings and infrastructure along our coasts, we're going to have to continue to place sacrificial sand on our beaches. Where we can, we're building structures to try to reduce erosional hotspots, but we've got agencies that go through a very rigorous permitting process, and I can attest to that process from both the public sector and the private sector, where we run complex computer numerical models to evaluate how that sand is going to equilibrate on the beach and where it's going to end up to ensure that the sand doesn't damage our reefs once it's placed on the beach, even if a hurricane comes through and pulls all that sand offshore.
Thank you, Penny. Do we want to answer? Yeah, go ahead.
We got a question about stony coral tissue loss disease, asking for more information on the selection and protocols for outplanting coral fragments that survive stony coral tissue loss disease. For example, are the fragments treated or quarantined, and are they outplanted or is only outplanted in only diseased areas? So I think, do we still have, do we want Alex to take that one, or Joe? Are you still with us?
Yeah, I can start off with that one. So I can only speak obviously to what CRF is doing, and it's important to note that, you know, the way that our restoration program works, all of our corals are in situ always. You know, our nurseries are offshore in the ocean. Our restoration sites are obviously very close to those nurseries, so there's no real need to do any kind of quarantining before putting them back in the same water. What we will do, though, is constantly monitor in our nursery the species we have in propagation that we're growing that are susceptible to stony coral tissue loss disease. And if we encounter any colonies, they're immediately removed from the nursery and sort of sacrificed for the benefit of the rest of the stock. And then we will similarly, after outplanting, monitor our corals in the months after to assess, you know, the presence of any stony coral tissue loss. And again, if we encounter any, those fragments are removed from the reef and sacrificed so as not to spread to the adjacent colonies. This has been very successful for us to date. We haven't really seen significant outbreaks of stony coral tissue loss, either in our outplanted coral populations or the natural corals in the areas where we're outplanting them. So we've been very lucky in that regard, but we're always vigilant on that front because obviously it's a disease that moves very quickly and can decimate a population if it's allowed to take hold.
Thank you. We have any, I think that addressed it. Looking through the questions here. We've got the online Bleach Watch training classes, the link is here in the chat if anybody's interested in that, and that'll be distributed. What else we got? Do we have anyone that wants to ask a question directly?
Yeah, this is Valerie Paul from the Smithsonian. I wanted to ask Alex a question. During your presentation, Alex, you indicated that some of your OFABs and other things that are in your nursery no longer exist in the wild, presumably because they just died from disease. So do you see less prevalence of disease in your nurseries than you would on adjacent reefs? And if so, do you have any thoughts about that?
Yeah, that's an excellent question. Thanks, Valerie. So yeah, I mentioned that it's not actually just the OFABs, Orbicella fabulata, species of boulder or star coral. It's actually all of our corals. There are representative genotypes, strains in our nursery that are no longer found in the wild. And yes, that's because the parent colony has died off due to disease or, you know, bleaching, random chance, anything like that. We do see lower rates of disease outbreak in our nursery and bleaching as well because the corals are, when they're grown on coral trees, actually removed from a significant number of stressors that they encounter naturally on the reef. So when you think about the water is more or less the same, right? So you have the same salinity levels, the same acidity levels, the same water temperature, obviously, between the reef and the nursery setting. But when you grow the corals suspended from a coral tree, it removes the prevalence of sedimentation, of algae overgrowth, and also predators that actually feed on the coral. Two of the primary predators for our coral species, at least, are yellow-footed snails and fireworms. And those animals just don't live in the coral trees, really. And so when you remove the coral from the substrate, from the bottom of the ocean, and put it on a coral tree, there's less algae overgrowth because we're constantly cleaning and maintaining the trees. There's obviously no sediment that is choking the corals in the middle of the water column, and you've removed some of those key predators. So we see much lower rates of coral death, coral disease, bleaching, things like that in our nursery. And what that also means is that the corals can grow much more quickly when they're suspended because they're not investing energy in securing a really firm base on the reef substrate, and they're allowed to grow in sort of 3 dimensions instead of just unidirectionally up the way that they do on the reef.
Thank you. Seeing some more questions here about sand grain size. I know a lot of people have concerns about beach nourishment. We all do, but again, we can't stop development along our barrier islands, and that's the only way we'd be able to get around beach nourishment at this point. But there is testing done of the sand that's placed on the beach, in addition to the geotechnical characteristics. One of the things that we find even when a condo is doing an excavation for a new pool deck is that there's naturally occurring high levels of arsenic on the beach. And that naturally occurring high levels of arsenic that's in those sands that are excavated from existing properties can't be placed back on the beach because it doesn't meet the state standards. And so a lot of those sands, when people are developing within the coastal construction control line, have to be replaced with sand from an inland mine. And so in the past, a lot of that material was excavated and placed on the beach, but now more and more of it is tested before it's placed in the beach dune system. You know, again, we want to protect our, not only the corals and what's offshore, but we also want to protect the constituents that are using the beach and enjoying our beaches, our wildlife, and our tourists. What else do we have here? I think we've covered most of the questions from the chat. Again, more information will be available for future webinars. I did see a webinar request for stony coral tissue loss disease updates. We can certainly consider that. We were talking about having more webinars if these continue to be popular. And with that, I think I'll hand it back to Alyssa.
Okay. Well, I want to thank Penny for moderating and all of our speakers today. Thank you. This would not have been possible without you. And I did notice we have a couple commissioners that are also in our participant list. So I wanted to thank Beam Fur, who spoke earlier, Commissioner from Hollandale Beach, Sabrina Javiana, who's on the line as well, and Commissioner Carol Shuham from Hollywood. Both of them will actually be moderating our next upcoming Ocean Day webinars. Commissioner Shuham for the Marine Wildlife webinar and Commissioner Javiana for the low nutrient landscaping and gardening webinar. So I wanted to thank everyone who showed up, everyone who stuck with us through the technical difficulties, every sponsor, all of the organizing partners, all of the elected officials who have come to get educated about coral reefs and are champions of them in their cities. So thank you everyone for coming. I will be sending out an email in the coming day or so with the recording. I'll try and put the two together. Any questions that didn't get answered that we can have the presenters type up answers for, organizations and how to assist, as well as the diver links such as Cfam and whatnot. So without further ado, thank you all for joining us and caring about coral reefs and listening to us for an hour and a half with technical difficulties. We are grateful to you and have a great day. Thank you, everyone.